Dr. Hongkui Zeng from the Allen Institute for Brain Sciences. Dr. Zeng received her PhD in molecular and cell biology from the Brandeis University, where she studied the molecular mechanisms of circadian clock in the fruit flies. Then she did a postdoctoral fellow at the MIT, where she studied the molecular and synaptic mechanisms underlying hippocampus-dependent plasticity and learning. She joined the Allen Institute in 2006, and then she became the director and executive vice president in 2020. Since then, she has been leading the structure science division and other programs as well. Her current research interests are in understanding neuronal diversity and connectivity in the mouse visual cortical circuit, and how different neuronal types work together to process and transform visual information. She has been recognized by multiple awards, such as being a member of the National Academy of Sciences in the US, and also the NIH Beacon Award. She has also presented lectures in Society for Neuroscience, IBRO, and Organization for Human Brain Mapping. And she has an H index of 83 with more than 36,000 citations and 156 scientific articles in journals. I wasn't going to say magazines. Journals like Nature, Nature Neuroscience, Cell, and Neuron. And she's also an advisor of the an advisory board member for Cell and Neuron. So I appreciate our distinguished members coming here today. Thank you.
Yeah, can you hear me well? Okay, thank you very much to Eduardo for inviting me to give a seminar today, and also thanks for that very kind, generous introduction. It's a tremendous pleasure here to give the talk, because this is really the first scientific presentation I give ever in Mexico. And I such an honor to be on this beautiful campus and interacting with all of you, and I very much look forward to more interactions with the scientific community in Mexico, between Allen Institute and Mexico. Okay. Okay, so with that, today I will tell you about our work at the Allen Institute for Brain Science in understanding the diversity and organization of cell types in the mammalian brain. The collective goal of neuroscience is to understand how brain works, at multiple levels, from the components to computation to the ultimate generation of behavior and cognition. We know that this is a daunting task given the extreme complexity of the brain and the nervous system. With millions to billions of neurons and a similar number of non-neuronal cells in the nervous system across a wide range of species. I know that many researchers here studying the different kinds of species, not just mice, but fish, mollusks, non-human primate, and human brain itself. And those millions to billions of neurons form intricate connections, trillions of connections, highly complex circuits. So in order to tackle this complexity, we think that it's essential to take a multi-level approach, from the basic component of cell types to how they form functionally specific circuits, to the in vivo activity of the different cells and how they correlate with the generation of behavior, and eventually perhaps to a computational understanding of how the brain processes information and generate behavior and cognition. So at the Allen Institute, we're also taking this kind of a systematic approach to investigate the brain at multiple levels. And we do this through two distinct neuroscience units, which I want to tell you about here today. The first is the Allen Institute for Brain Science, which was the oldest institute in now the parent Allen Institute. And we just celebrated our 20th anniversary last year, so it was started in 2003. And the primary focus of our institute, the Brain Science Institute, is to understand the molecular and anatomical foundation of the brain, focusing on understanding the diversity and cell types and wiring diagrams. Basically, we call it the parts list and the maps of the brain. And the second institute, which was recently established in 2021, but also inherited a MindScope program from, which started 10 years ago. And this is Allen Institute for Neurodynamics. They collaborate with brain science very closely, but they focus on studying circuit activities, behavior, and computation, using a number of experimental approaches, including multi-scale electrical recordings using, for example, neuropixels, probes, optical physiology, including large-scale calcium imaging and other neurotransmitter imaging, structure-based brain-wide activity mapping in collaboration with us, and working on behaving animals in different paradigms, and also at computational level using AI ML-based approaches to help with the analysis of the data as well as the generation of series, computational series about how circuit works. They also have a program called OpenScope, which is like a brain observatory, which we open up our imaging and recording capabilities to the communities to have open calls of proposals to invite researchers, selected proposals, to come to the institute to generate data using our capabilities. And of course, at the Allen Institute in general, we very much practice this kind of a big science, team science, and open science approach, making all our data at all these different levels publicly available through our online website, and allow people to use our data to integrate with researchers' own data and perform joint integrated analysis and a deeper understanding from the data. So my talk today will be mainly focusing on the work that we do in the Brain Science Institute, really focusing on understanding the cell types and the wiring diagrams. So as we all know, ever since the pioneering work of Ramon Cajal more than 100 years ago, there are many different types of neurons and non-neuronal cells in the brain exhibiting tremendous diversity. And this diversity is reflected not only at the morphology level, as beautifully illustrated by Ramon Cajal himself, but also at multiple other levels, from gene expression to anatomy to even spatial location, and to the physiology and the connectivity of the cells. So it is really essential to profile the cellular properties at all these different levels and understand how cell types, how the cells can be grouped or classified into types, and how the different types of cells are organized in the brain. So in order to tackle this challenge, at the Allen Institute, we have developed multiple technology platforms to profile individual cells and perform data-driven discovery, use the data-driven discovery approach to understand the classification of the different cell types. Our front-end approach is the highly scalable single-cell genomics technologies. And those technologies include single-cell RNA sequencing or single-cell transcriptomics, single-nucleus attack-seq under the umbrella of epigenomics, as well as the spatially resolved transcriptomics at a single-cell level. And we use those approaches to profile hundreds of thousands to millions of individual cells and generate the first iteration of the classification, the taxonomies and atlases of the cells in the brain. And then we continue to create genetic tools to target those specific cell types using the molecular information we obtained from the genomic studies. Traditionally, we generate transgenic driver lines using specific marker genes to label cell types. Of course, that's a very labor-intensive and expensive process. So in recent years, we have switched into using viruses to incorporate cell type-specific enhancers that we mined and obtained from the combination of single-cell RNA-seq and single-nucleus attack-seq data, and testing those enhanced elements in the AAV viral vectors. And with a pretty good successful rate, we are able to identify specific enhancers that can drive fairly specific cell type targeting in the brain. And we can now do that, not only in a traditional model organisms such as mouse, but also apply those enhancer viruses into non-genetic model organisms, including non-human primate, and even in the human brain itself. At the anatomical level, we use large-scale light microscopy, fluorescent light microscopy, as well as electron microscopy to understand the brain-wide projection patterns of the individual neurons using light microscopy and the synaptic level connections of the local circuits at the electron microscopy level. To integrate molecular information generated from the omics data sets and anatomical and connectivity information generated from the imaging data set, we use a set of multimodal integrative approaches, including a triple modality patch-seq, barcoded connectomics, in which we use barcoded AAV virus, injecting into a particular brain region. And by in situ sequencing, we are able to decode the exon projection patterns of the labeled neurons and relate that with the transcriptomic profiles of the individual cells that contain the same barcode. And we also combine traditional anterograde and retrograding, again, with molecular profiling, such as retro-seq or retrograde coupled with spatial transcriptomics to, again, relate gene expression information, the transcriptomic identities, with the long-range input-output projection connectional patterns of the individual cells. Underlying all these different technology platforms, we build strong computational capabilities that will allow us to not only manage the tremendous amount of data that we generated, but also allow us to perform a large-scale analysis, especially cross-modality integrative analysis, and for the creation of the cell type atlases. And also generating the online web platform that will allow us to disseminate, to display the data, and build visualization tools to allow the community to use our data and knowledge and tools gained from the study. Okay, so with this overview, now I will tell you a series of studies, mostly using the front-end single-cell transcriptomics approach, starting from a single brain region, going all the way to cover the entire brain to see what we have learned through the process. So noting that our study is very much in a kind of a hypothesis-free, data-driven approach, we think this kind of approach that we are taking at the Allen Institute is very complementary to what's normally done in the academic setting, where individual labs are focused on particular hypotheses, using a hypothesis-driven approach to do deep investigation of a particular problem. We think that both approaches, this kind of a horizontal, large-scale data-driven approach and the deep vertical hypothesis-driven approach are highly complementary to each other, and both are needed in order to understand the complexity of the nervous system. Okay, so in this first study, I will tell about, this is actually a collaborative effort within the BRAIN Initiative called the BRAIN Initiative Cell Census Network, which we are a part of. And in the network, we want to create multi-model cell type atlas. And the first region that we focused on is the primary motor cortex. So here shows the atlas that we created of all the cell types in the motor cortex across 4 mammalian species, from mouse to macaque monkey to marmoset and to the human itself. So we not only creating an atlas, importantly, we also can extract organizing principles or rules of cell types in the brain. And those rules are, I should say that they are also very consistent with a number of other studies published individually around that same time. So those rules are, cell types are organized in a hierarchical manner, as shown in this hierarchical tree, a dendrogram, and that are divided into, that are defined into different, with different levels of classification of cell types, from class at the top branch level to subclass to type. indicating a different level of granularity of the relationship between the different cell types. We are able to computationally integrate all these different characterizations together, so multi-model integration, including transcriptomics, epigenomics, spatial distribution, morphology, projection, and physiology. And we find within this hierarchical organization a coexistence of discrete and continuous variations of all these different kinds of cellular properties, with discrete distinctions among major classes and subclasses at the top branch level of the tree, whereas many different kinds of continuous variations at the terminal leaf, the type, or the cluster level. Similarly, we also observe the coexistence of cross-species conservation and divergence of those cell types. Again, the conservation mainly exists at the top level, class and subclass level. For example, we are able to identify an invariant of 28 subclasses of neuronal, glutamatergic, GABAergic, and all the non-neuronal types. And the number of 28 subclasses exist across all the four mammalian species we studied. The divergence across species mainly exists at the cluster or type level, where we rarely find specific one-to-one correspondence of individual clusters. It's very often this kind of a fuzzier multiple-to-multiple correspondence. And we also observe a substantial degree of gene expression variation. For example, about 20% of the genes in the genome would exhibit different expression patterns across species, even among the clusters or the subclasses that are conserved across species. So altogether, those different rules paint a complex cell type landscape. We cannot say specifically exactly how many cell types are in a particular region, but this kind of organization is a nice summary that provides both the generality as well as the specific situations under different conditions. And you will see a lot more as I go on. So, okay, the second question we asked then is that this is coming from one cortical region. How do cell types compare across different cortical regions? Are they the same or different? So in this next study in the mouse, we profiled using single-cell transcriptomics 2 different cortical regions, anterolateral motor cortex in the front end of the cortex and the primary visual cortex at the back end. And we generated a similar hierarchical tree with about 130 cell clusters identified with both regions together. And the major finding from this study is a very interesting dichotomy between the 2 different classes of neurons. The GABAergic inhibitory neuron types at the cluster level, they are all similar, common, or shared between those 2 very different cortical areas. The excitatory glutamatergic neuron types, on the other hand, are highly distinct between the 2 different areas. Each of the transcriptomic cluster contains cells coming predominantly only from one of the 2 regions, rarely coming from both regions. And also we observe a large number of differentially expressed genes between for each glutamatergic cluster between the 2 different regions compared to GABAergic neurons. So this dichotomy, we hypothesized that this dichotomy between glutamatergic and GABAergic neurons in the cortex may be due to their differential developmental origins, with the excitatory glutamatergic neurons developmentally generated within the cortical plate itself, in which the cortical plate is subject to morphogen-regulated cortical gradient that eventually leads to the formation of cortical areas. The GABAergic neurons in the cortex, on the other hand, they were generated from the subcortical ganglionic eminence areas and migrate into the cortex during development. And as such, maybe in their developmental, or intrinsic origins, they don't carry this kind of a region-specific transcriptomic signal, gene expression signal in their transcriptomes. So then next, we further scaled up our effort from profiling 2 different cortical areas to a study that covers all all the cortical areas, as well as the neighboring hippocampal formation regions of the mouse brain. Basically, more than 30 individual cortical areas covering the entire dorsal surface of the mouse brain profiled. And from this, we generated a large taxonomy, of course, again, hierarchically organized. Without getting into the details, I just wanted to say that the main finding from this study is that we find, again, in a glutamatergic cell type class, we find a highly distinct yet a parallel organization of all the glutamatergic cell types between the cortex or the isocortical or the neocortical region and the hippocampal formation, which is considered a more ancient, evolutionarily more ancient cortical structure. So, so the distinction, is shown that all the hippocampal formation clusters are shown in this kind of a greenish color in this UMAP versus the kind of rainbow-colored cortical neurons from different regions that are all combined together. You see that they're distinct, but they also show a certain homologous relationship. And this homologous relationship can be revealed by searching for the isocortical subclass homologs for every single hippocampal formation or HPF cluster. And you see this kind of correspondence. Is actually quite specific here. So in summary, putting all the different glutamatergic transcriptomic clusters together, we identified 240 clusters, a very large diversity. We organized all the different clusters based on the brain, the regions where they come from. Isocortical areas on the left, hippocampal formation areas on the right, and hippocampal formation includes, just by the way, includes the hippocampus itself, including CA1, CA3, dentate gyrus areas, as well as the neighboring subicular complex and lateral and medial entorhinal cortex. So they are organized by regions, but they are also organized by their layer distribution. Cortical areas are organized in this laminar structure with 6 layers. And superficial to deep, we see a amazing correspondence in terms of the laminar specificity and their potential long-range projection patterns between isocortex and hippocampal formation glutamatergic types, suggesting that even though those two different regions are highly distinct, evolutionarily separated for millions and millions of years, their cell type organization, there's this highly corresponding cell type relationship that is conserved in their transcriptomic profiles. Okay, so with this study, we then further moved on, scaling up our single-cell transcriptomics profiling again, now covering the entire mouse brain. So, and this work, again, coming from the Brain Initiative Cell Census Network, the BICCN. We now call it a BICCN 2.0 project because it was really a capstone completing project, for the consortium. And in this consortium, we worked closely together with 6 other major labs in the US in generating multi-model single-cell genomics data set, including, so the lab names are listed here. The data modalities include single-cell and single-nucleus RNA-seq of millions of cells profiled, and also single-nucleus epigenomic data sets, including a methylation-seq and a tag-seq. And also, finally, it also includes a set of 3 different sets of whole brain scale single spatial transcriptomics data set using the MERFISH technology, as well as the slide-seq technology. So the different technologies are complementary to each other. The coverage of the whole brain is all shown here. We covered all the major brain structures in telencephalon, forebrain, telencephalon, diencephalon, midbrain, hindbrain, and cerebellum. In the study that I just told you about, only covers 2 of the major brain structures, isocortex and hippocampal formation. And now we have all the other structures covered. So it's a tremendous experimental undertaking as well as a computational undertaking. And then now I will only tell you about the work that we have done at the Allen Institute in using the combination of two brain-wide scale data sets, a single-cell RNA-seq and a MERFISH, creating a high-resolution transcriptomic cell type atlas for the entire mouse brain, and what we have learned from that. Okay. So here's the eventual taxonomy and atlas that we have generated, as visualized in this UMAP that contains all the millions of single-cell transcriptomes that we used. So here I can tell you a little bit about how this atlas was generated. It was actually quite an interesting undertaking. So using the 10x genomics single-cell RNA-seq method, we profiled 7 million cells across the entire mouse brain, covering all the different regions. And out of that, we established a very stringent QC process in which we failed 3 million of the 7 million single-cell transcriptomes, leaving behind only 4 million high-quality single-cell transcriptomes that we used for clustering analysis. This is high quality is really important for us because our goal is to generate a reference atlas for the community. As you can understand, the reference atlas really needs to be high quality because then any investigators can map their own RNA-seq data, and maybe the quality is not as good or as stringent as what we have, but nonetheless, they will still be able to map their data into the high-quality reference data in order to obtain an unambiguous or less ambiguous cell type identity. If your reference is already low quality, then the mapping result would not be good. Okay, so that's why we have 4 million cells left. So using those 4 million cells, we performed iterative clustering to obtain as many clusters computationally allowed. And we obtained more than 5,000 clusters for the whole brain out of the 4 million cells. And then from those more than 5,000 clusters, we extracted differentially expressed genes that can distinguish every pair of those 5,000 clusters. We obtained a list of 8,500 differentially expressed marker genes. And then from those 8,500 differentially expressed marker genes, we selected the top 500. Top 500 marker genes that collectively have the greatest discriminating power in differentiating all those 5,000 markers. And then we used those 500 top 500 marker genes to perform the brain-wide MERFISH experiments. Okay, so this particular data set is coming from a single mouse brain and from using those 500 genes. Very high quality data. And after, again, quality control and segmentation, we obtained 4 million segmented MERFISH cells from that one single data set. And then we mapped each of those 4 million segmented MERFISH cells using these computational approaches, map back into our taxonomy of 5,000 clusters that was derived from the single-cell RNA-seq dataset to obtain a cell type or cluster identity for each of the MERFISH cells. And then finally, conversely, using the spatial location of each of the MERFISH cells belonging to a particular cluster, we assign the spatial identity for the transcriptomic cluster. Okay, so through this kind of an iterative process, we generated a taxonomy of 5,322 clusters, each with a specifically assigned spatial location. Many of them highly specific, beautiful spatial location, which you will see later. So that is our final version of the cell type taxonomy and atlas. And from there, we then also grouped the different clusters into different hierarchical levels because we're not sure that all 5,000 clusters are true cell types. And we really wanted to understand the relationship between those 5,000 clusters. So we grouped them into 1,200 supertypes, 338 subclasses, and 34 classes. We further group them into 7 anatomically based neighborhoods that will allow us to do further analysis, but the neighborhood is not part of the hierarchy. So, okay, so all these different definitions of cell types are shown in this UMAP. And in this particular slide, the UMAP is colored by the 34 classes, subclasses, and it's also colored by the regions where the cells come from, that is assigned based on the Murphy data, and also by the neurotransmitter type each cell type has, especially the neuron types. We find that a major finding from this work, just initial finding from this work, is the extraordinary diversity of the neuron types in the brain. So neurons form 5,200 of the 5,300 clusters that we obtained through this kind of unbiased sampling. Whereas non-neuronal cells only compose about 100 clusters. So this, using a uniform clustering method, we reveal this extraordinary diversity in the neuronal populations across different parts of the brain. Our second finding is that we found that for the neurons, the cell types are not only identified, defined by their molecular identities, for example, by what kind of neurotransmitters they express, glutamatergic, GABAergic, or other modulatory neurotransmitters such as dopamine, but they are also very much defined by the regions, the regions where they come from. So in fact, the classes and subclasses that we've defined here are driven by 2 factors, molecular identity and regional specificity. Okay, so on top of that, just very briefly, we are now also able to, once the cell type clusters are defined, we're able to extract all kinds of gene modules. And we focus on the transcription factor gene modules. We identified 52 transcription factor co-expression modules. And we find that those transcription factor co-expression modules exhibit high degree of specificity at all different hierarchical levels, class, subclass, all the way down clusters. And they define the relationships of the different types in such a specific organized manner that we think that these modules form collectively form a combinatorial transcription factor code that can define the cell types and their spatial localization across different parts of the brain. This includes both neuronal and non-neuronal cells. Okay, so then if we go just down a little bit further to try to understand, just get a sense of the specificity, I use this one slide to show the example that we go one step deeper and we still see this amazing diversity and spatial specificity. So here I show 3 of the 6 neuronal neighborhoods that I mentioned, all in the forebrain region. So this is all the glutamatergic neuronal cell types that are defined in telencephalon or the forebrain. We call it also called a developmental structure called palea. And this one shows all the GABAergic neuronal types that are present in telencephalon because they were generated in the subcortical regions, which is also called subpalea. That's how we call them. And finally, all the neuronal glutamatergic and GABAergic neuronal types that are found in hypothalamus and its closely related extended amygdala areas. Each of those neighborhoods contains hundreds to thousands of individual clusters and with exquisite spatial specificity revealed by the Murphy data. And I just go down one further level. I take one, we're doing a lot of studies and trying to understand the diversity within each of those different major brain structures. I use the subpallium GABA as an example to reveal the next level of organization in this slide. So this contains all cells coming from the subpallium GABA neighborhood that generates all the GABAergic neuron types in the telencephalon. Okay, and they are, without getting into details, they are organized into 7 classes, 52 subclasses, 284 supertypes, and more than 1,000 individual clusters. And the spatial location of each of those clusters is revealed. And the organization, it's really everything, it just makes sense. GABAergic neurons coming from, that are located in the cortex, their relationship with the GABAergic neurons, both local interneurons and long-range projection GABAergic neurons that are found in striatum and a major basal ganglia region. And also many of the new GABAergic neurons that we didn't know much about before that come from the pallidum, the ventral pallidum areas, are all shown here in a well-organized manner that makes sense. And we also observe regional difference. Just very quickly mention the regional difference. We find that the cortical GABAergic neurons often exhibit this kind of continuous variations, maybe because they are evolutionarily newer. They haven't segregated that much. But you can see this kind of continuous variation in the UMAP shown here. Whereas the subcortical, the basal ganglia, or the striatal and pallidal areas, the GABAergic neurons are showing in this highly distinct, highly complex, tiny little, many tiny little clusters, as shown on the other side. On the other side of the UMAP. As you can see, this side of UMAP and this side of UMAP, very different. It reflects the different properties of the individual clusters that we identify. Okay. So also wanted to show another kind of diversity, and this is extracting out all the neuronal cell types that express modulatory neurotransmitters. So this is taking out the cells coming from different parts of the brain, put them together into this UMAP. It's very different from the previous UMAP I showed. And the neurotransmitter types that we identified are shown all here. We identified all the cholinergic, dopaminergic, serotonergic, noradrenergic, and histaminergic neuronal types across the brain. And we find, very interestingly, they show a heterogeneous degree of transmitter co-expression patterns. So very often, the modulatory neurotransmitters, such as dopamine, is not only is expressed or secreted just by itself in a particular cluster. Very often, it's co-expressed or co-released with glutamate and or GABA. So this co-release pattern is very prevalent, at least in our transcriptomic data. And I will show, go a little bit deeper in the next slide. But here, I just want to highlight among all the different neuromodulatory cell types we identified, this is the major subclass that forms the midbrain dopaminergic neurons. And this is the major subclass that composes of the midbrain and hindbrain serotonergic neurons. And we can go deeper in here. The next slide. This is the single subclass that we identified containing only 4,000 cells. But it contains all the dopamine neurons and its related, the relatives, within the substantial nigra, VT8, and midbrain raphe areas. And those cells within this one single subclass can be further divided transcriptomically into 8 supertypes, as shown by different colors, and 43 clusters. I mean, this kind of molecular heterogeneity is just, it's not invented, because you can see it just with the UMAP. And also, we superimpose the neurotransmitter content each of the cluster or supertype has, and they are shown here. There are cells that can express or release dopamine only. The ones that co-release GABA, co-release glutamate, or co-release both GABA and glutamate, or not. There are even GABA and glutamate cells that do not express dopamine that belong to this particular subclass. So high degree of heterogeneity, and how are they related to the spatial localization? So this is the correspondence between the UMAP and the Murfish data. We can see the segregation, right? Spatial segregation, especially at the supertype level, is pretty clear. This brownish, reddish supertypes is mainly present in the substantial nigra, right? So, and then the others are mixed together within the VTA area and also within the midbrain raphe, dorsal and medial raphe areas, showing a high degree of heterogeneity. And when you go down to cluster level, of course, there's even more. The heterogeneity is mind-boggling. So this is, you can generate lots of hypotheses. We stop here because there will be much more work going forward in understanding the physiology of the different dopaminergic clusters we identified here, and how they are related to their long-range projection patterns of those dopamine neurons that go into, from substantia nigra, VTA, and Raphe nuclei, how they go into other parts of the brain and how they interact with other cell types. Much work is needed. Okay, so I'm not sure about time, so I'll just keep going, okay? So I told you about this extraordinary diversity of neuronal types, and not so much in the non-neuronal type. That's kind of a little bit disappointing, but the non-neuronal types will come back later. They are also very interesting. But we still observed some very interesting diversity in the non-neuronal types, and especially in the astrocytes and the ependymal cells. So I used this one slide to show the interesting diversity we observed. And this is the UMAP showing all the astrocyte clusters we identified throughout the brain. And they are corresponding spatial distribution. The spatial transcriptomics data is so critical for us to understand why the cells are organized like that transcriptomically. So we find that there are 3 major groups of astrocytes in the brain. The dominant group is, of course, the astrocytes that are present in the gray matter that are intermingled with neurons together. And those dominant group of astrocytes are organized in a region-specific manner, from olfactory bulb to telencephalon regions to non-telencephalon regions, and go all the way back to cerebellum. So you see this correspondence of clusters with the spatial specificity. The second kind of astrocyte is this special one called interlaminar astrocytes. And those are the astrocytes that line in the PO surface of the brain and send their processes into the gray matter. And then the third group of astrocytes are the ones that are lining, that's related to adult neurogenesis. Those are the special kind of group of astrocytes that align this rostral migratory stream, which is a special organization, a structure of, that generates, that perform adult neurogenesis of factory granule cells, GABAergic granule neurons, generated from the subventricular zone in the dorsal part of striatum, and migrate into the olfactory bulb through this migratory stream. And this migratory stream is formed by this tunnel of astrocytes, and which by themselves also have potential neurogenic potential according to the literature. What we observe here is that this astrocyte clusters, that form this kind of stream in the transcriptomic space, perfectly correlating with the location of each of those clusters from the dorsal striatum subventricular zone all the way into the striatum. So there's this, again, beautiful correspondence between transcriptomic specificity and spatial specificity. The second example is the ependymal cells. Ependymal cells are very similar to astrocytes, and they are the special kind of cells that line all the ventricles, kind of a neuroglial type of cells, but they line all the ventricles in different parts of the brain, and they are important for brain-blood interaction or brain-cerebral spinal fluid interaction, important for nutrient sensing, metabolite sensing, and the regulation of hormone and nutrient exchange between the brain and the body. So we identified many different subclasses and types and clusters of ependymal cells, and each of them has this beautiful spatial specificity, lining a particular ventricle, medial, lateral, anterior, posterior parts of the brain. And one particular example is this tenicyte subclass. Multiple clusters of tenicytes lining only the ventral third ventricle contained within the hypothalamus, and are known to be important to have multiple morphological and physiological subtypes that are important for nutrient sensing and exchange for the hypothalamic and metabolic function. So we now have transcriptomic clusters corresponding to them. Okay, so this is like the general description of about what we have learned from this gigantic whole brain cell type atlas. Here I just wanted to say that in conjunction with the publication, we now have built a new online platform, which we call Allen Brain Cell Atlas, or ABC Atlas, in which we display the entire single-cell transcriptomic data set and the spatial transcriptomic morphage data set that allow users to mine and find out where their favorite cell types are. Furthermore, we also have developed a computational tool called Map My Cells. It's a tool, online tool, that allows user to upload their own single-cell transcriptomics data or even spatial transcriptomic data, upload and then map each cell of their own data into our reference atlas to obtain a cluster or cell type identity for the user's data. And you can compare that identity with your own clustering result, of course. But this provides a standardized reference that allows different researchers to be able to compare with the reference and then compare with each other as well. Okay. Okay, so in the next remaining few minutes, I want to also just now talk about some unpublished work, just going beyond, atlas generating, telling you about how we are utilizing this data to understand how cell types change, under different conditions, in particular, development and aging. And also how transcriptomic cell type identities correlate with physiological or connectional properties. So just a few studies, very quick slides to show you. So in this particular study, we studied using single-cell RNA-seq to profile cell types in the aging mouse brain. Okay, so yet another study with about 1.2 million cells, half from the 18 months old mouse, right? And the young adult, which is the same data set that we used to build the whole brain atlas. We sampled 16 broad regions covering about, in the forebrain, midbrain, and hindbrain, covering about 1/3 of the entire brain. And we also generated spatial transcriptomics data to validate the findings we made in this study. We used the Map My Cell tool to map all these different cells, the aged cells, transcriptomes, into our reference atlas to obtain the cell type identity. And we did also de novo clustering as well to try to identify age-specific clusters. So overall, in this data set, we identified about half of all the subclasses that we defined in our whole brain atlas, 172 subclasses, and about 850 clusters. We used different computational approaches to reliably identify age-associated differentially expressed genes. And out of this, from this data set, we identified a large number of age-associated differentially expressed genes, 2,500 of them, and also a small set of age-specific clusters. So as you can see here, this kind of changes between young adult and aged mice, there are not so many age-specific clusters, meaning that the transcriptomic profiles are not changing dramatically. Only in special cases, like microglia, age-associated microglia, for example, you see highly distinct new kinds of cells emerging or different cell state emerging. But in vast majority of the cases, you see the cell types are still there, but their gene expression profiles have shifted. Okay, so then this shows all the 2,400, 2,500 HDE genes, their distribution across the brain, different brain regions. And non-neuronal supertypes are shown here, and neuronal subclasses are shown here. And also, this shows a dot plot showing like how many of the DE genes are present in each of the supertype or subclass, for example. A major conclusion you can see here is that there is a lot. So even though non-neuronal cells don't exhibit much diversity, they exhibit a lot of changes, age-dependent changes. So many of the HDE genes are associated with all different kinds of non-neuronal cell types, including astrocytes, oligodendrocytes, ependymal cells, as well as vascular and immune cells, microglia and things like that. On the neuronal side, we see major changes in the cortex. And also, interestingly, we see major changes in hypothalamus, which I will show you later. Okay, so in this one, in this particular map, we highlight the changes in non-neuronal and immature neuronal types. And with all these ones highlighted, we find, as I said, widespread gene expression changes associated with age in all the different classes and subclasses of non-neuronal cells. And in particular, also major changes associated with immature neuronal populations and astrocyte populations that are associated with adult neurogenesis. We see dramatic reduction of adult neurogenesis in different parts of the brain, including dentate gyrus and the olfactory neurons that I told you about. On the neuronal side, this is a summary of all the HD genes we identified. We see a cloud, this gray cloud of HD genes that are present in many, many, many neuronal subclasses across the brain, but the changes are all relatively small. They concentrated on this low side. But then at the same time, we also identify a small group of neuronal subclasses that exhibit major changes. And strikingly, among those small group of neuronal types, we find a large number of hypothalamic neurons. Hypothalamic neurons that exhibit dramatic age-associated changes. So this is a summary diagram showing the kinds of changes that we see on a global level. For the aging brain, we see overall decreased adult neurogenesis, decreased neuronal structure and signaling, especially in the structural integrity is the axon projection integrity. That's reflected by both in both neuronal side as well as in the oligodendrocytes. As you know, oligodendrocytes form this sheath covering the neurons, the neural axons. So any changes associated with oligodendrocytes indicating the deterioration of the axon fiber tracts. Simultaneously, we also see dramatically increased immune responses in almost all of the non-neuronal populations, especially microglia, and BAMs, which are border-associated macrophages, as well as in vascular cells. Increased immune responses. We even observe increased immune responses in some specific neuronal populations. And that goes to this middle point, the intersecting middle point. Between neuronal responses and immune responses, we find an aging hotspot, which is exactly located in the hypothalamus, especially areas surrounding the ventral third ventricle. And the cell types involved in this hypothalamic hotspot for aging, including the specialized ependymal cells that line the third ventricle, and in particular the tanycytes I told you about that line the third ventricle, as well as a highly specific group of neuronal types that are important for energy homeostasis as a major hypothalamic function. And they are shown in this slide here. This is the third ventricle, and this within hypothalamus, and this shows the ependymal cells lining the dorsal part of the third ventricle, and the different tanycyte subtypes that are lining the ventral part of the third ventricle, as well as the median eminence area that are known to be important for energy homeostasis. And you see the large number of gene expression changes specific to those two types compared to astrocytes. And this shows all the major neuronal types, and not even at the subclass level. Now it's going down to specific cluster level, highly specific hypothalamic neuronal clusters that express specific neuropeptides and hormones that are known to be important for homeostasis. This shows the spatial location of the hypothalamic clusters that are changed under aging conditions, including those within the arcuate nucleus, and also dorsal medial nucleus, and also paraventricular nucleus. So the location of them shown here, and the changes are shown here, number of the genes, as well as whether they have the increased immune response or not. And here shows the marker genes for those clusters. Well-known neuropeptides, AGRP, MPY, POMC, CRH, the stress hormone, AVP, OCT, oxytocin. So they are just popping up, and including also leptin receptor cluster and GLP-1 receptor, the latest anti-obesity drug So the specific clusters are changed in the aging mouse brain. Okay, so now I'm running over time. I'm at time now. So just quickly move down to several other studies. This is one slide showing now we are also studying the development of cell types. And we use the mouse visual cortex as a particular example. In this data set, single-cell RNA-seq densely sampled across the entire prenatal and postnatal development of the mouse visual cortex, starting from embryonic day E11.5 all the way to P28, and then adult. And with this dense sampling, we are able to obtain the trajectories of the development and differentiation of the cell types that are defined in the adult stage. Without getting into the details, I just wanted to mention one interesting observation. And in the future, I will talk about this work in more detail. So here, I want to highlight the perinatal time point, and E18, 18.5 to P0. And so that's the perinatal junction between embryonic development and postnatal development. All the major cell subclasses, all the neurogenesis happens during the embryonic stage. And after the pup is born, it's really mainly just a differentiation, neuronal differentiation, and forming the circuit, activity and circuit and emergence of the visual response properties of all those neurons. We know a lot about the neurogenesis, how the diversity happens, but we actually know very little about the cell type differentiation in the postnatal stage. So this time point is shown as this part in the UMAP. Okay? It's right at the perinatal junction. Before that is neurogenesis, generating of the major neuronal and non-neuronal cell classes. After that, you can just simply see from this UMAP, there's actually tremendous diversification that's happening after birth, postnatally. And that's in conjunction with all the events that are happening with the circuit formation. So So the major conclusion from this part is that postnatally, there's actually tremendous cell type diversification that goes hand in hand with the circuit formation and maturation within that circuit. So yeah, a lot to be understood. And now we have the molecular handles, molecular signatures for all the transcription factors, adhesion molecules, ion channels, synaptic proteins. When they emerge in what specific cell type, we have all that data information now that can lead to a lot of hypothesis-driven studies into understanding exactly how that cell type differentiation happens. Okay, so I want to finish now just very quickly. So we also have connectivity study. Everybody knows the different cortical areas and brain regions have extensive connections forming networks, modules, and networks, and different cell types are involved in the formation of those network connections. And very much all of them at the population level. But how do these inter-area connections are related to cell types, transcriptomically defined cell types, and how they are related even at a single cell level? That's a major question in neuroscience that we may be able to address now. And in this published study, we used the single cell morphology reconstruction approach to obtain the long-range projection patterns of about 1,700 individual neurons from cortex, thalamus, and also striatum. And what we find is that major molecular cell types, each type has highly distinct long-range projection patterns. So that's kind of confirming the validity of the molecularly defined cell types, right? So that's good. But on top of that, in addition to that, we also observe extensive heterogeneity among the individual cells, even within the same molecularly defined cell types. And those additional heterogeneity is reflected in three aspects: regional specificity, topographic specificity. There's tremendous amount of topographic specificity within the brain. And also individual cell variation. Each cell chooses a subset of the target that its type exhibit. So something more that we need to understand. And finally, we use this patch-seq multimodal method to try to bridge the transcriptomic information with morphologies and with electrophysiological information. And in the first example study that we did was to profile cortical GABAergic interneurons that are known to be tremendously diverse, within a particular cortical region, from mouse to human. So this is from mouse visual cortex, cortical GABAergic neurons, and we used the patch-seq approach to classify them into 28 what we called MET types. So those 28 MET types are actually less diverse than the 61 transcriptomic types, or T types, that we mentioned. They are less diverse, however, they show better cross-modality consistency. So, okay, and those 28 MET types are defined and consistent with traditionally defined GABAergic neuron types, parvalbumin and somatostatin positive, VIP positive, and LAMP5 positive, which is the nucleoform cells in layer 1, and also this is SNCG positive, which is what people usually call the CCK positive basket cells. So the MET types have intermediate granularity, less granular than the transcriptomic types, but they have high cross-modality concordance and mutual predictability between transcriptomic profiles and morphoelectrical properties. They also recapitulate traditionally defined GABAergic interneuron types well, and with further subdivision that's very often driven by layer specificity. And finally, we think that the MET types, the MET types, can serve as a Rosetta Stone to bridge molecular and morphoelectrical properties in completely disparate, separate data set. And that's shown in my last data slide here. So we used the patch-seq data for the GABAergic interneurons to infer molecular identities of the neurons that we reconstructed from our electron microscopy data set. So in the electron microscopy data, all the information we have is the morphology of the cells that we reconstruct, and of course, their local synaptic connections. So in this particular example, we are able to use the morphology as a common descriptor for cell types in the EM space and in the patch-seq space, and then to link transcriptomically defined cell types with their specific local circuit connectivity patterns. And we observed a good degree of specificity, actually, local cell type specificity of the synaptic connections for 3 major somatostatin transcriptomic types. Layer 4 targeting, layer 5, superficial layer 5 targeting, the intrathalamus-cephalic targeting, and also the cortical, subcortical projecting layer 5 neuron-specific targeting somatostatin types. Okay, so with that, I want to finally, the ultimate goal for us, this is only the main work that we've done, a lot of the work we've done in mice. But we are also now trying to expand that together with many of our colleagues at the Brain Initiative Consortium to obtain a comprehensive cell type map across species and try to bridge cell type information with brain function. So we want to do this through the creation of brain-wide cell type map across 4 dimensions. Across species, we're now doing similar kind of extensive profiling in human and non-human primate brains, especially in generating single-cell transcriptomics data, single-cell epigenomics data, and spatial transcriptomics data, even in those larger brains. And we want to do it across modalities. Of course, as I mentioned at the last few slides, it's important to understand what does all those transcriptomic types mean? How do they relate to the structure and function of the cells? Across space, we don't want to just look at one brain region. We want to look at all the different regions in the brain. As you know, many of you study particular circuits. You know that circuits, they work together to generate behavior. And the information is passed along in the different circuits. So you need to understand the cell type organization across different regions. And finally, across time. And that's where the dynamics come from. The transcriptomic map is only a static one-time view, snapshot of how cell types look like. But they change. They change dramatically throughout development, as I showed, and they also change in aged conditions. And furthermore, they change in different functional and diseased conditions. So this is really a starting point for us to begin to understand how cell types and their function change under a variety of different diseased conditions. Okay, with that, I don't want to get into the details of the summary of the major scientific conclusions that I told you about. I just wanted to thank a really large team of scientists and investigators and leaders at the Allen Institute in making this kind of a collective, large-scale effort happen. We have project teams, and those listed the leads, project leads of each of the project teams, and our scientific leaders at the top. And it's really this kind of collective, multidisciplinary collective work, from experimentalists to computational scientists to technologists, to put everything together and disseminate all the information that we have. Thank you for your attention.
Now we have some questions. Hugo?
Thank you. Well, this is just mind-blowing science that probably are going to change many of hypotheses of how the brain works.
Yeah.
I'm amazed about the heterogeneity of cell types based on your transcriptomics.
Yeah.
That do not match necessarily with the morphological or the physiological subtypes. So I'm wondering what's your hypothesis regarding why you need so much diversity at the molecular level when the anatomy and the physiology is similar?
Yeah, why do we need so much diversity? I don't know why, but that's why the brain is so capable of doing so many different things. I think this diversity that we observe is just, it's more than, I think it's going to be magnitudes of more than any of the other organs in the body. The brain is just special. And it's all set out by this plan, the transcription factor code I told you about. And this kind of distances between different transcriptomics clusters we identify, the distances are just bigger. They are bigger than when you go into liver or kidney or whatever. That lays out the plan, for all the things that the brain can do, in sensation, behavior, and also the regulation of the entire body, right? So, and I don't think the transcriptomic diversity is unique. You will see similar or maybe even more heterogeneity and diversity at the morphology and the connectivity level, right? Once, someday if we have a connectome, right? If we understand how each cell is connected with each other, there may be even more. For example, I just say, when I give talk like this, and the neuroscientists who study the mouse or human, they say this is too much. It's just like, how can there be so many types, right? 5,000 types. But when I talk, but the Drosophila people, fruit pie people, they often come to me to say that I'm not surprised at all, because in the Drosophila brain, we already have the connectome. We even have thousands of cell types based on connectome in the Drosophila brain. Of course, the mouse brain would have more. So it's there, and we're just beginning to scratch the surface.
Thank you. Any other questions? Luis?
Hi. That was very, very exciting, of course. And I have a related question.
Yeah.
So if you have a bunch of cells that share a lot of features, it is intuitive that they're going to end up in the same anatomical place. But the opposite is not as intuitive. When you have neighboring cells that are very, very different in a lot of aspects. And at the end, you sort of alluded to the glial cells and how they make them end up where they are. But I wonder if there's something intrinsic in these neighboring yet distinct cells that make them end up in the same place.
Yeah, that's a very interesting question. That's why we're now studying development, right? We want to see how the plan of the brain is laid out. There are highly distinct cells that are right next to each other. So does that just define a border between two domains? right? Or maybe the cells, they migrate, from beta, they come together. And I think both exist. So now we can see the heterogeneity within a particular brain region, and also between different brain regions. That heterogeneity is because the same group of cells that just diversify, as I mentioned, in the mouse visual cortex development, they just diversify like that, or actually they come from different places and they come together. It's both possible. And now we want to, so spatial transcriptomics is so important, we want to trace, where that comes from.
Thank you.
Okay, any other questions? Otherwise, I have one. Oh, yeah, Luis, the other Luis.
Yeah, Luis.
We have many Luises here.
Hi, thank you for your talk. So you briefly showed that the molecular identity of the neurons kind of determine the connectivity, but do you think that if you have the transcriptomic profile of a neuron or two different neurons in different brain areas, can you predict that they will be connected?
Yeah.
Is someone doing that already?
Yeah, I mean, there are people who are trying to do that, and now we have the data. Yeah, so I'm not sure that if that's the case, but we can look into it. Whether I think in some cases that may be true. Cells, maybe it's like a homophilic, right, attraction that leads to the cell types that are connected to each other. They express the same gene, they find each other, and they form connections. That's possible. But there could be also, there's this, in addition to the transcription factor code that I talk about in terms of defining cell type identity, now we can begin to look at adhesion molecule code, or whatever the kind of molecule code that defines the connectivity between different kinds of cells. Yeah, I cannot endorse a particular hypothesis yet, like what you're saying, but that, that's related to data mining and a further validation experiment.
Right, thank you.
Yeah.
Okay. Yeah.
I have a very similar question.
Yeah.
I'm just wondering if the population patterns of activity will influence the types of cells that you are identifying. I'm meaning that different types of cells involved, for example, in fast rhythms in different parts of the brain will
Fast rhythms.
Yeah, will be sharing transcriptomic profiles.
Yeah, well, I don't know, possible. Yes, well, that's another experiment, right? So whether it's transcriptomics defining it or it's really just a correlation. I think because gene expression is really underlying auto function, right? In order to have a function emerge, you have to express specific genes at the right time that enable that particular activity, right? So I can imagine that there could be a set of genes, like ion channels or something, that allow you to have this kind of a fast spiking or rhythm, right? That generate this kind of rhythm. And that same set of genes, that gene module would be reused by a different cell type that will allow you to generate the same kind of physiological properties. Yeah. It's possible. So that it all is like once you have the data, you can find the corresponding genes.
Thank you.
Yeah.
Thank you. I have a more technical question.
Yeah.
So can you briefly mention how people usually use the tools that you make available for everyone for their own research? Like how can students or researchers here use the tools?
Yeah. So the tools are maybe just specific examples about the cell type tools that we generate. This will facilitate a lot of the integrative analysis. The different data generated from different groups, there may be batch effect, experimental variability. So you need to be able to integrate with our data, but also different data, you need to use correct computational methods to normalize the data, harmonize them together in order to generate, to identify meaningful changes, right, in your condition. So using our reference data will help you with that. And also, if you have some data of a particular cells, but you don't know exactly where they are, you can go do the mapping and go to our spatial transcriptomics data. So then you can find out exactly where your cell types are. You can also, if you know a particular brain region that you are working on, you want to find out what cell types are in that region, you go to our website, you have the list of cell types, and you have all the gene expression profiles for those cell types. that you can use for your study. Maybe you can design experiments for knockouts, for other kind of studies to further understand what the cells do.
Thank you.
Yeah.
There are no more questions. I want to thank our visitor for her presentation. Thank you.
Thank you very much. Yeah, okay. Overwhelming, yeah. So, yeah, it's a lot.